The next speaker is Rebecka Sepela. She's a postdoc at Harvard University. She studies octopuses, and she was unable to pick a favorite fact about octopuses, but many of the things she likes about them are the high level of RNA recoding, the interesting mating behaviors, and the fact that they have really cool copper binding abilities in their blood.
Yes, well, thank you for the introduction, and thank you all for your attention. I am Rebecka Sepela. I'm a postdoctoral researcher in the lab of Nicholas Bologno, and today I'll be discussing how octopus taste by touch chemosensation is mediated by environmental microbiomes. In the Bologno lab, we're interested at the intersection of evolution and sensation, namely, how do organisms evolve within the context of their niche? How does the ecological niche drive evolution? And in the face of rapidly changing environments, how do sensory systems cope? To begin to ask any of these questions, we must first characterize the sensory niche of the specific animal that we're studying. And while this idea seems simple, it's actually quite nuanced. And I like to put up this quote for reference. Organisms living in the same place at the same time inhabit different worlds. They live in different sensory environments bounded by the properties of their sensory organs. This concept is encapsulated by the idea of the Umwelt, a term coined by a German biologist in the 1900s. Within the Umwelt, we have all of the abiotic components of an environment, as well as the biotic ones. And it's worth considering how the animal interacts with these. I would like to highlight what I think of what might be an underappreciated component of the Umwelt, and that is microbes that exist at the interface between the abiotic and the biotic world. If we are to understand how animals sense their world, we must consider this world to be a microbial one. For 3 billion years after bacterial life originated, animals diverged from their protistan ancestors into these seas teeming with bacteria and have lived in close association ever since. Now, with beautiful studies of symbioses, we are becoming more and more aware of these intricate associations, whether it be from the Vibrio that allow these squid to fluoresce and camouflage, or the Candidatus that allow the mouthless deep-sea tube worms to produce energy in an otherwise inhospitable environment. This interkingdom signaling is well appreciated in internal symbioses. But we want to know, are external cues also leveraged in transient interkingdom interactions? And to ask this question, I'm using the octopus as a model. The octopus is a beautiful benthic animal that lives in a world covered by microbes. It's using its 8 arms to touch and interrogate every surface that it touches, sensing what's on there. Now, the way the octopus senses its world is through these sucker cups that have specialized chemoreceptors in their sensory cells. And in the Bologno lab, we've been studying these chemoreceptors and how they've diverged from a nicotinic acetylcholine receptor-like ancestor. These CRs are pentameric ion channels that, in the presence of a ligand, will gate a nonspecific cation conductance and inform the animal that they have sensed a relevant ligand. Now, despite being related to a nicotinic acetylcholine receptor, the octopus CRs have lost this tightly coordinated ligand binding pocket shown up here and are no longer able to bind neurotransmitters. Instead, the octopus CRs have adopted this really broad, really hydrophobic, flat ligand binding surface. This hydrophobic surface allows the octopus to bind sticky molecules, such as the terpenoids that we find in our freezers. Now, we think this is especially relevant for an animal that lives in an aqueous environment where it's wanting to touch surfaces and understand those surfaces. But the question at hand, what chemicals have the octopus CRs actually evolved to sense within the ecology? So to answer this question, we thought about what surfaces the octopus needs to sense. We started off with thinking about their prey. Octopus are voracious hunters and opportunistic scavengers. They need to differentiate viable prey from something that might be decaying or potentially toxic, and they need to do this without sight. Additionally, female octopus are meticulous brooders, where they will spend the last little bit of their life's energy continuously aerating and grooming their eggs. Curiously, they'll occasionally kick out eggs from their clutch. Do either of these predation or brooding behaviors rely on microbe-facilitated chemosensory cues? To answer this question, we started by profiling the microbiomes of these different surfaces, and we see that indeed the microbiomes on living and decayed crabs are distinct, as well as the microbiomes on mothered and rejected eggs. But to really get at whether or not these communities are producing sentient chemical cues, we had to work with the microbes. So for that, I cultured microbes from these surfaces of interest, grew them up on solid media, isolated them in liquid culture for sequencing and validation, filtered them into supernatants, and applied those supernatants onto Xenopus oocytes that were expressing our octopus receptor. I would then rank these supernatants in their ability to activate the receptor and compare that to an uninjected oocyte control. And what we found is that there were certain select strains of bacteria that were able to activate this octopus receptor, and through large-scale cultures, aqueous organic phase extraction, as well as fractionation with HPLC, we're able to iteratively screen different fractions of these supernatants to identify single chemicals that would constitute this bioactivity with mass spec and NMR. I've done this protocol for a couple of different strains of bacteria, some strains isolated from crabs, as well as some strains isolated from eggs, and I was able to pick out two distinct classes of molecules, one being this beta-carboline from the crab-derived bacteria, as well as these flavin-like molecules that we found from the egg-derived bacteria. Now, with these single molecules in hand, the question is, are these relevant to the octopus? So what we could do is apply these molecules in a plume of seawater to the octopus arms, and we see that similar to a positive control, the crab and the egg-derived molecules will cause arms to autonomously move and also elicit peripheral nerve activity, and that's in contrast to the application of a vehicle control. We next use structural biology to look at how these different environmental ligands would associate with the receptor, and maybe to no surprise, but we found that these molecules, regardless of their origin, associated within that super sticky hydrophobic ligand binding pocket. Now, what was a surprise is when we looked a little bit closer, we saw that our crab molecules and our egg molecules assumed different binding poses within that pocket. The crab molecules were able to utilize this beta nitrogen to form a hydrogen bond with the serine on the channel. And we see that in both of these different crab-derived molecules, whereas our egg molecule mainly bound by hydrophobic interactions. Using electrophysiology, we could manipulate these, either the hydrogen bond acceptor on the channel or the hydrogen bond donor on these molecules, and indeed confirm that this hydrogen bond interaction was important specifically for the crab-derived molecules and not those of the eggs. So, do these different binding mechanisms mean anything for the animal? To assess that, we looked at another part of our structural biology work, and we found that the crab molecules, when bound to the pocket, would facilitate the pore to assume a conformation that was not especially permeable to calcium, whereas the egg-bound structures would flip in this glutamate that would help coordinate calcium and help push it through the pore. And once again, we could confirm that with electrophysiology, when these octopus receptors were bound with egg molecules, they were passing higher levels of calcium, suggesting that even in the most peripheral sense, the octopus could distinguish chemicals and produce distinct cellular signals. So next we wanted to know what these cellular signals actually mean to the animal. To get at that, we went back to our microbiome data and looked at where the crab-producing strain of bacteria localized, and we saw that was enriched in decaying crab communities. Interestingly enough, our molecule would glow blue under fluorescence, and we see that these decaying crabs are also quite blue. Granted, that's highly correlative, but what's maybe more convincing is that we could use mass spec to detect this crab molecule from decayed crabs. And similarly, we could do an analysis with our egg bacteria, which was enriched in rejected egg communities and also prevalent by mass spec within those environments. So then the final test of what do these chemicals mean was something that we could assess with octopus behavior. So this was kind of fun, but what we did is we took these little toy crabs and let the octopus interact with them. If the toy crabs were doped in our DMSO control, the octopus would readily investigate it, touch it, and then kind of envelop it into its buccal web and kind of just hang on to it. These cute, curious creatures. But if the toy crab had been doped with our molecule from the decaying crab bacteria, the octopus would go over and touch it, and then choose not to engulf it. And we could do this many times over and quantitate that. And we found, similar to a aversive positive control, these crab-derived molecules would bias the animal not to engulf it compared to our seawater controls, somewhat recapitulating how the octopus behave with true decayed crabs. And we could also do a similar sort of experiment with our egg-derived molecule, where we give brooding mothers who are sitting in their den these little agar eggs that are either doped with a vehicle control or our egg molecule, and we could count how long she would tolerate holding them. And if the egg molecule is present, the octopus would kick it right out of her clutch. So in summary, through this project, we've exploited the microbial ecology of the octopus to identify pertinent sensory cues. And we have found that beta-carbolines from decaying crabs and flavins from rejected eggs will inform predation and breeding behavior by binding octopus CRs and biasing different signal transduction or gating modes. The results of these studies suggest that even transient interactions with microbes inform animal behavior, which is something that we expect to be prevalent across the entire tree of life, even from C. elegans that will avoid pathogenic microbes to this beautiful example in single-celled choanoflagellates, which will form these multicellular communities upon the happenstance interactions with different microbes. Mind you that these choanoflagellates are our closest relative, closest single-celled relative. So I would like to argue that understanding how microbial cues are leveraged will help us inform the animal experience and eventually understand how symbioses evolve. And then I'd like to end the talk with one last idea, and that being, since microbes are a fast adapting biotic interface to the abiotic world, and I believe that we can exploit them to understand how the world is changing around us. So with that, I'd like to thank you all, and thank you all for your attention, and thank my collaborators at Harvard, as well as UCSD for their integral work to this project. And I'll welcome any questions.
Thank you. I'm sure there's a ton of questions. I will go first. So you've shown that adding it to a decoy crab or decoy egg would get the octopus to reject it. Could you block the receptors and show that that blocks the rejection response, so that really these receptors are what's responsible?
That's something that we're working on. Right now, we don't have any pharmacological tools that will allow us to do that, but we're in the process of doing high-throughput calcium screening-based experiments to try to find some, because that's such a great question. Mm-hmm.
Awesome. Thank you. Right over there. I think Angie's closer.
Hi, great talk. So I can see how a bacteria that was eating or decaying a crab would signal that it might be toxic, but is the one that is found on the eggs have any relation to the egg viability that you know of? Like, is it, if the egg were taken care of, would it survive, or is this some bacteria that is deadly to the egg and could infect the other ones if it's not kicked out?
That's also a great question, and unfortunately, this egg rejection is quite difficult to study. It happens quite randomly. The best we can do is look at the microbiome right now and see that this strain of bacteria that produces these molecules is enriched in rejected eggs. We know that once the eggs are kicked out of the clutch, they will not continue to develop. So we don't know if they're kicked out because they're spoiled or if they're kicked out for another reason. But once they are kicked out and we retrieve them, they do have this unique microbiome profile, and that is distinct from an egg that we can prune from the clutch and just let it sit in the water. So there's a difference between mothered eggs, rejected eggs, and also intentionally unincubated eggs. It's an interesting question that would be cool to study for.
Yeah, very cool. Very cool. Can you Do you have any more details maybe about how the selectivity for related molecules to the one you tested, and B, other bacteria that might produce the same compound? To have an idea about specificity of this. Is it like something specific for that bacteria, or you tried other bacteria might produce similar things and it might react the same way?
Can you say again?
So on one hand, molecules that might be related, how specific are for those molecules versus related flavins or something like that?
Oh, yeah.
And B, other bacteria that might elicit similar responses.
Okay. So on a receptor level, with the beta carbolines, there's a really nice structure-activity relationship where the better that beta nitrogen is at accepting a hydrogen bond, the stronger effect it has on opening up the channel. With the flavin molecules, since we don't have that hydrogen bond to analyze in a structure-activity relationship, we can modulate how hydrophobic they are, but really quickly we run into this limit where they're too hydrophobic and they start crashing out of solution. So that relationship isn't as tight. Beyond the receptor level, we can go and look at structure-activity relationships at the nerve and the autonomous arm movement level, and we see that generally the chemicals that are best at causing the arm to move or the nerve to activate are these ones that we find in nature, and not necessarily synthetic analogs that are closely related in a structural sense. Does that answer your question?
Yeah, and we can follow afterwards.
Okay, thanks.
Well, thank you.
Yeah.
Okay, maybe one quick question if somebody has an urgent one. No? Okay, great. Then we can move on. Thank you, Rebecka.
Thanks.